Welcome to SciFlux. Here, you get daily coverage of the latest scientific research. Every day, we bring you new research in a clear and simple format so you can stay informed without wasting time. Like the video, subscribe, and leave a comment to stay connected and help this channel grow. And if you want each new research delivered directly to you, join the SciFlux newsletter. This paper presents the green synthesis of zinc oxide nanoparticles, ZNONPs, using Acmella caulirhiza, merging traditional medicinal knowledge with sustainable nanotechnology. An aqueous leaf extract facilitated environmentally friendly zinc oxide nanoparticles formation. Characterization confirmed crystallinity, X-ray diffraction, spherical morphology, scanning electron microscopy, stability, 31 mV zeta potential, and an average size of 97 nanometers, while FTIR identified the functional biomolecules driving synthesis. Next, the study assessed bioactivity. The ZNONPs showed strong antibacterial effects against Staphylococcus aureus, 17 mm, and Escherichia coli, 15 mm, over 50% antioxidant activity, significant anti-inflammatory properties, and low hemolytic activity, 12.68% at 500 micrograms per milliliter, confirming safety for biomedical use. Overall, this work demonstrates a sustainable route to highly bioactive zinc oxide nanoparticles, highlighting the integration of traditional plant wisdom with modern nanotechnology for environmentally responsible medical applications. This paper explores strategies to uncover risk genes across the full spectrum of genetic variation, addressing the missing middle of intermediate frequency variants often overlooked by current methods. While rare, high-impact variants are detected through family studies and common low-impact variants through large GWAS, intermediate variants remain largely invisible. These hidden contributors play critical roles in conditions from psychiatric disorders to cancer. Next, the study reviews innovations to bridge this gap. Advanced variant annotation, joint modeling of multiple genetic signals, refined phenotyping, and network-based inference to map gene interactions. By treating the full frequency spectrum as a unified axis, researchers can systematically reveal previously hidden risk genes. Overall, this work enhances our ability to identify disease-associated genes across all variant types, moving toward a more complete understanding of human health and the genetic architecture of disease. This paper explores a graph transformer autoencoder with latent diffusion for capturing and analyzing the human brain's complex network dynamics. Traditional functional brain graphs often examine connectivity, activity, or topology in isolation, missing interactions between these features. This AI framework compresses full brain graphs into a low-dimensional latent space, guided by spectral geometry to ensure biologically plausible representations, and reconstructs the original graphs from this compressed form. Next, the study highlights its capabilities. The latent space distinguishes working memory states, decodes visual stimuli, captures temporal neural dynamics, and generates synthetic yet biologically realistic brain graphs. Overall, this approach provides a unified, interpretable representation of brain connectivity, offering new opportunities to study neural patterns, model disease-specific networks, and guide personalized interventions with AI-generated, structurally accurate brain graphs. This paper explores the intersection of neuroscience and artificial intelligence, examining how principles from the human brain can inform more efficient and adaptable AI systems. Modern deep learning has drawn inspiration from neurons, attention mechanisms, and network architectures, yet it remains fundamentally different from biological learning. AI struggles with incremental knowledge accumulation, biologically plausible error correction, and adaptive generalization. Next, the study reviews how recent discoveries in synaptic learning, neural coding, and dynamic information processing can guide the design of AI that learns more like the brain. It identifies both successful translations and remaining gaps, highlighting opportunities to improve learning efficiency and flexibility. Overall, this research suggests that bridging AI with the brain's mechanisms could yield systems capable of adaptive, generalized understanding, moving closer to human-level intelligence by leveraging the biological blueprint already within us. This paper explores improved modeling of the COVID-19 reproduction number, R0 and RT, addressing challenges posed by presymptomatic transmission. Accurate R estimates are critical for tracking epidemics and guiding interventions. Traditional models rely on the serial interval, the time between symptom onset in infector and infectee, but presymptomatic spread can produce negative intervals, complicating calculations. Standard approaches like the Renewal and Lotka-Euler equations often become inconsistent when negative serial intervals are unrestricted. Next, the study introduces a refined model that imposes a realistic lower cutoff on negative serial intervals. This adjustment reconciles differences between modeling methods and provides more reliable reproduction numbers. Overall, the research ensures epidemiological models produce consistent, accurate R estimates, supporting better-informed public health decisions on vaccination, restrictions, and reopening strategies during pandemics. This paper explores a novel evolutionary strategy, the replacer phenotype, which optimizes reproduction by targeting individuals of a different type, eliminating wasted reproductive effort. Traditional Moran processes allow chance to nullify reproductive events when an offspring replaces the same type, slowing adaptation. Replacers, by contrast, strategically ensure each reproduction influences population composition. Next, the study models replacers in well-mixed and structured populations. Even a single neutral replacer sees its fixation probability rise from 1/n to roughly 1 square root of n. They can overcome deleterious mutations, resist invasion by new mutants, and dominate populations even with significantly lower intrinsic fitness. Overall, this research highlights that in evolution, efficiency and strategic action can outweigh raw reproductive output, suggesting that clever, targeted behavior may be as crucial as strength for trait success. This paper explores TRAECR, an end-to-end pipeline designed to pre-process and harmonize PET imaging data for precise, reliable analysis. PET scans provide critical insights into conditions like Alzheimer's and cancer, but raw data is noisy, variable, and labor-intensive to prepare. TRAECR, template registration, MRI PET co-registration, anatomical brain extraction, and combat ravel harmonization transforms these complex datasets into clean, standardized, and comparable formats. Next, the study highlights its workflow. Scans are aligned to a universal brain template, co-registered with MRI, and corrected for scanner or site variability. This standardization reduces errors, removes batch effects, and ensures consistent, high-quality data across studies. Overall, TRAECR streamlines PET analysis, enabling faster, more reliable evaluation of therapies, improved disease monitoring, and deeper understanding of neurological and pathological processes, accelerating discovery and enhancing clinical insight. This paper presents a plug-and-play TEER upgraded organ-on-chip system for real-time monitoring of biological barrier integrity. Biological barriers, like gut linings, blood vessels, and lung air sacs, are critical for health, but traditional TEER measurements are complex, provide only snapshots, and require specialized expertise. This new system integrates fixed, semi-transparent TEER electrodes directly into the dynamic 42 organ-on-chip, enabling continuous, non-invasive monitoring inside the incubator without disrupting microfluidics, multicellular colonization, or imaging capabilities. Next, researchers validated the platform using a 3D intestine-on-chip model, tracking barrier formation, disruption, and recovery. Real-time measurements demonstrated higher sensitivity than conventional endpoint assays, capturing rapid cellular dynamics. Overall, this innovation simplifies and enhances barrier monitoring, providing continuous, high-resolution data that can accelerate drug discovery, improve understanding of host microbiota interactions, and advance research on pathological conditions. This paper explores a dynamic role of the nuclear periphery in gene transcription, challenging the traditional view of Lamin B1 regions as transcriptionally silent. Using mouse embryonic stem cells, researchers discovered that a subset of active genes, primarily essential housekeeping genes, are transcribed directly at the inner nuclear membrane. Remarkably, as stem cells differentiated into olfactory precursor cells, the number of these periphery-associated active genes increased. These genes are GC-rich and regulated by zinc finger transcription factors, highlighting specific sequence and regulatory features. Next, the study demonstrates that the nuclear edge is not merely a storage site for silent heterochromatin, but a dynamic hub of active transcription, redefining our understanding of nuclear organization and its role in fundamental cellular processes. Thanks for watching. If you enjoyed the video, please like, comment, and subscribe. Also, join our newsletter to stay informed with the latest discoveries shaping science and innovation. See you tomorrow with a new round of breakthroughs. Take care